Dear ESAG conference attendees, I'm pleased and honored to talk about variant interpretation in colorectal cancer syndromes. This talk will summarize work performed under the umbrella of INSIGHT, the International Society for Gastrointestinal Hereditary Cancer. The issue of variant interpretation has been present since the genetic bases of disease were discovered and the first genetic tests, starting from karyotype analysis, became available. With the advent of molecular analysis and the unveiling of an unprecedented number of genetic variants, the issue has become more and more burning. Initially, the problem has been dealt with using subjective assessments that have led to the development of qualitative pathogenicity criteria. However, since the beginning, it was realized that this approach is error-prone and that standardized and objective rules are needed to achieve reliable and clinically valuable interpretations of the huge amount of genetic variation that is detected in the diagnostic laboratories. Initially in our group, we focused our interest on the mismatch repair genes. Here you can see some of the themes that are still recurrent nowadays in diagnostic labs and among clinicians who are in charge of conveying their results to patients. The variants that are more commonly problematic in the clinics are those that can give theoretically rise to missense substitutions or small in-frame deletions duplications. For the MMR genes, the parameters that were considered at the time were the phenotype of the patient family, co-segregation of the variant with the phenotype, allelic population frequency, physicochemical properties of the newly introduced amino acid compared to the wild type, and degree of evolutionary conservation in ortholog genes. These variables are usually considered for all genes investigated in the diagnostic labs, and potential mismatch changes still represent a major challenge, although we know that all types of variants are potentially pathogenic, and therefore the interpretation criteria must consider all potential types of variations. Mistakes in variant interpretation can be detrimental to patients, as they can lead to over or under treatment. Paradoxically, they can also be advantageous to the community. This is what happened with one of the first two papers that reported the cloning of the first Lynch syndrome gene, MSH2. The study identified two families carrying the same intronic variant, C2006T2C, according to current nomenclature, while the nucleotide at the time was number 2020. Co-segregation with a phenotype was observed. This variant has been soon after classified as benign or class 1, since the minor allele frequency attains polymorphic values in the population. The two families harbored another pathogenic variant that was not discovered at the time. This story tells us that we have to be careful in relying only on clinical data and that a functional confirmation should be obtained, which was not the case in the Cell paper since the presumed effect on RNA was not investigated. Most of this talk will be dedicated to the interpretation of gene variation in MMR genes. The work performed on these genes and on Lynch syndrome is an example of a successful initiative that has been and is still ongoing and has evolved over several years, especially after the involvement of INSIGHT. It has also represented a widely appreciated model for the interpretation approach to other cancer and non-cancer genes. Lynch syndrome and the MMR genes have been particularly attractive for the interpretation exercise for several reasons. First of all, since their discovery as disease causative, testing for these genes is frequently requested to several laboratories. The estimated prevalence of Lynch syndrome, the condition caused by their malfunction, may be as high as 1 in 279. These observations show that variant interpretation is a common clinically relevant problem. The functioning of the MMR machinery is quite well known, at least the mechanisms involved in the repair of polymerase errors in base incorporation and replication slippage. Consequently, functional assays have been developed for MMR activity and other components of MMR protein function, with the production of a conspicuous amount of data. Importantly, characteristic tumor markers of MMR deficiency are available, and they are commonly tested in diagnostic labs. An intriguing feature of the MMR tumor tests is that they are a sort of in vivo functional assay, since they provide indirect information on DNA repair activity in tumor cells. Finally, the proportion of VUS that are identified in diagnostic tests is high. About 30 to 40% of the variants reported in the INSIGHT database and in ClinVar are still of uncertain significance, meaning that approximately one-third of the families with a suspicion of Lynch syndrome or with a mismatch repair gene non-polymorphic variant still lack a clinically useful test report. These numbers further demonstrate the urgent need of a solution to this problem. INSIGHT entered the interpretation stage in 2007, 2 years after its establishment from the merger of the Leeds-Castle Polyposis Group and the International Collaborative Group on HMPCC, when the first embryo of what has later become the Variant Interpretation Committee met spontaneously at the biennial conference in Yokohama, Japan. The first actions concerning the variant database were undertaken at that time. The original MMR gene database had been established in 1994 within the frame of the ICG HMPCC by Hans Vasen and Päivi Peltomäki, who was at the time the curator. This became the INSIGHT database when INSIGHT was founded in 2005. Additional databases, one literature-based, set up by Mike Woods in Newfoundland, and a functional database run by Rolf Simons and others in the Netherlands, were developed shortly thereafter. The three databases, INSIGHT, Canadian, and Dutch, were then merged in a single one that is publicly accessible through the INSIGHT website, building the infrastructure for the classification work. The database has then been upgraded and is currently part of the shared global Variome LOVD database. The merger required a big data cleaning effort to amend imprecisions in nomenclature, duplicate entries, and different origins of the variants, among others. This has led to the identification of 2,360 unique constitutional variants on which the group has focused for the initial interpretation. The first official step of the Interpretation Committee has been the decision to adopt the classification system proposed by the IARC Working Group on Unclassified Genetic Variants, chaired by Sean Tavtigian in 2008. This is comprised of 5 classes: pathogenic, likely pathogenic, uncertain, likely non-pathogenic, and definitely non-pathogenic, that are distinguished by cutoff probability values derived from multifactorial analysis. The system was initially conceived based on the BRCA1-2 experience, for which multifactorial analysis has been widely applied. For other genes, including the MMR genes, it was recommended that while tools for multifactorial analysis were under development, qualitative approaches be developed through a consensus process involving a panel of experts. Importantly, the system has been conceived for the classification of high penetrance variants of well-established clinical utility, while other approaches should be devised for moderate and low-risk variants. I already mentioned the need to evaluate 2 independent sources of datasets for an accurate and reliable classification. The example of the original MSH2 paper in Lynch syndrome explains why clinical information cannot be considered as a standalone criteria. On the other hand, we know that functional assays have variable specificity and sensitivity. The system was developed within the frame of a IARC project, and one of the main tasks of IARC is the evaluation of carcinogenicity of environmental agents. To this purpose, combined evidence from epidemiological studies in humans and from animal studies is evaluated. Likewise, for genetic variants, it was established that the interpretation should be based on combined evidence from clinical and functional data. For qualitative analysis, the following datasets were considered. Predictable sequence effects based on the laws of molecular biology. For instance, in most cases, nonsense variants can be assumed to be pathogenic simply based on knowledge of the DNA change, and no further data are needed in support of this interpretation. However, we do know that some protein truncating variants may escape this prediction due to their location in specific regions of the gene, such as in the very 3' region or in alternatively spliced exons. Clinical data include co-segregation analysis and type of family history. The phenotype of Lynch syndrome is highly variable. However, the finding of a variant in multiple families with a very strong phenotype is clearly more indicative of a causative association than the incidental detection in an exome analysis performed for clinical reasons unrelated to cancer. Tumor tests, MSI, BRAF, and MLH1 promoter status, and the MMR protein immunohistochemistry are a special subset of clinical data since they also provide indirect functional evidence. Frequency in control population is another important parameter, especially useful when a variant attains polymorphic frequency. Another peculiar characteristic of MMR genes is the phenotypic differences between monoallelic and biallelic carriers. The latter ones are affected by the condition named constitutional mismatch repair deficiency, or CMMRD, and have a more severe phenotype characterized by earlier onset, partially different tumor spectrum, and manifestations of type 1 neurofibromatosis. If you have to classify a variant and check in the literature or your dataset, you find that it has been observed in trans with a pathogenic variant in an individual affected with CMMRD, this is a point of evidence in favor of pathogenicity based on the assumption that the CMMRD phenotype manifests when two pathogenic variants located on different alleles of the same gene are present. Functional in vitro data are very important and are worth special considerations. Finally, computational in silico predictions can be a supportive point of evidence, and they can be useful for multifactorial analysis. There are several available types of assays that test in vitro different facets of MMR function. These include RNA and/or protein cellular assays. The latter can test overall repair activity or stability or subcellular localization of the single protein components of the system. Given the absence of standardization, the MMR Interpretation Committee decided that in order to consider a given assay for classification of a specific variant, concordant results had to be obtained by two independent laboratories. Of course, this reduces the utility of these assays, but at the same time, this approach was deemed necessary to avoid misclassifications due to lack of standardization of these tests. Based on combinations of the different qualitative datasets, the VIC has developed a set of classification criteria for the 5 categories envisaged by the IAC system. In addition, quantitative likelihood thresholds were considered using a multifactorial Bayesian approach devised for the MMR genes by members of the committee, namely Shantav Tigyan, Mendes Purdle, and Bryony Thompson. The multifactorial model takes into account characteristics of the sequence change, namely prediction derived from in silico tools and tumor data. Classification process has been performed using a modified Delphi approach with discussion in teleconferences of discordant interpretations following the assignment of variants to a set of reviewers. Discrepancies were discussed in teleconferences, which were also the setting to discuss the interpretation criteria. Using this approach, over 2,300 variants were initially classified, leading to clinically relevant category changes. For instance, as shown within the red circle on the lower right, nearly 5% of the variants that were previously deemed to be non-pathogenic were assigned to class 5 pathogenic. Conversely, 10% of previously benign classifications were assigned to class 5 or 4, definitely and likely pathogenic respectively, and these are inside the circle on the top left. Following the initial accomplishments of the Insight MMR Interpretation Committee, work has been going on with the accrual of further data, classification of additional variants, and update of the criteria. The classification criteria have been published along with reclassifications and have been posted on the Insight website. They are regularly revised and the different versions are visible for comparison. Criteria for the interpretation of functional assays have been deeply revised, and this is the flowchart showing the ideal sequence of tests that should start with RNA analysis, which, if negative for missense and in-frame indels, should be followed by MMR activity assays and then sequentially by other functional assessments in case no effect on repair capacity is observed. The need for further work is highlighted by the high fraction of class 3 variants or VUS that is present in the database. This is especially true for the predicted missense changes, 75% of which are VUS out of a total of about 1,200 variants of this type that are present in the database. The clinical utility of the classification system will be improved when highly reproducible and accurate functional assays will become available. In order to do this, for gene transcription analysis, the pattern of normal variations in the processed products of MMR genes must be determined beforehand. Traditional studies have relied on analysis of partial gene transcript fragments. However, co-regulated splicing patterns involving distant regions cannot be detected with these methods, nor they can provide accurate quantification of the different products. This requires analysis of full-length RNA by high-throughput sequencing, as shown by work performed on different cancer-predisposing genes, including the MMR ones, as shown in this paper by Monica Morak and colleagues in Munich. Still on the matter of RNA, our understanding of the implications of variations in 5' and 3' regions is lagging behind because only a few of these have been functionally tested. This scheme shows the paths that have been suggested following a review of available data. Allele-specific expression should be the first test to be performed. This is incorporated in the Insight guidelines for the evaluation of RNA effects, since it is a general transcription assay that can detect different types of abnormalities and quantitative thresholds can be established. More specific assays can be performed, but at this moment they are not included in the Insight criteria. RNA-seq looks promising also for this category of variants. The issue of reproducibility and accuracy is even more burning for protein and cellular functional assays. Different approaches have been proposed and are ongoing. The one shown in the top part of the slide is performed on normal cells from an individual. However, it requires a skin biopsy since it has been calibrated on fibroblasts while leukocytes have been found not to be suitable for the purpose. SIMRA on the bottom has the great advantage of being performed in a cell-free environment, and it is amenable to large-scale analysis. On the other hand, it does not assess other functional aspects such as protein expression levels or cellular localization. The performance of the multifactorial model is evolving through refinement of the estimates of Bayesian a priori likelihoods based on in silico predictions and improved incorporation of tumor characteristics. Combined use of these two features improves the performance of the model as assessed in this study on a set of 150 missense variants classified according to the criteria established by the American College of Medical Genetics and Genomics, ACMG, and the Association for Molecular Pathology. In 2014, ClinGen has assigned to the Insight MMRVIC the status of expert panel, which entails a high level of confidence in the classifications reported to ClinVar. However, ClinGen classifications are made according to the ACMG system, which has some differences with the IARC and Insight approaches, and other submitters may use their own criteria or adapt ACMG criteria to the MMR genes. This leads to discordances in the interpretations of variants contributed by Insight and other submitters, which in some cases lead to opposite classifications, pathogenic versus benign. ClinVar accepts and publishes variant assertions as they are, and the many submissions from multi-panel testing or whole genome analysis end up with variants of unknown significance as they do use an uncurated approach. On the other hand, Insight submissions are examined by a group of specialists in the field who may assign a different classification based on the available evidence. In addition, Insight also contains phenotypic information on individual patients, which further adds to the depth of information we have on each variant and aids in variant classification. Since it would not be productive to have separate guidelines for interpretation, which might also lead to divergence of the criteria and confusion for diagnostic laboratories, efforts are going on to harmonize the MMR interpretation guidelines, involving more actively Insight in the ClinGen frame, whose structure would also allow long-term viability of the Insight database. Insight, specifically John Paul Plazer, the database curator, has also developed a software for the interpretation of variants. The input is represented by the different datasets and the output is the classification. This has been tested in parallel with the classifications made during the teleconferences and has proved to work nicely. The software needs to be adapted to modifications of the criteria, including the changes that will be made to harmonize the interpretation with the ClinGen process. Insight is interested in all hereditary gastrointestinal cancer conditions, and its database contains sections for other non-MMR genes, including APC. So in 2019, the APC Variant Interpretation Committee was established. It is led by Stefan Aretz from the University of Bonn. Preliminary work made by Sherry In at the University of Melbourne has shown that also for this gene, the number and types of variants on ClinVar and Insight can be quite different. For APC on ClinVar, half of all variant records are VUS, while on Insight, 75 variants have a pathogenic interpretation, whereas variants of unknown significance only account for 5% of the total record. These findings call for additional levels of integration between ClinGen and Insight in the field of hereditary gastrointestinal cancer. On this matter, a proposal has been presented for a joint Insight ClinGen variant curation expert panel that includes two levels of interpretation, one based on the specific gene and the other one on the different types of evidence, with gene experts and evidence experts. In summary, these are the main lessons that we learned from the Insight effort in the last 13 years. Large-scale data sharing at a national and international level is important in order to achieve clinically useful interpretations. Therefore, it is very important that data submission is encouraged and possibly incorporated in the flow of diagnostic laboratories. Insight includes scientists and health professionals of various backgrounds who share a common interest in hereditary gastrointestinal cancers. These colleagues have gained experience in topics that were initially stranger for them. Geneticists are knowledgeable about GI cancer prevention and treatment. Conversely, gastroenterologists are acquainted with the genetic basis and molecular tests, and so many of them are able, for instance, to calculate recurrence risks for the GI cancer syndromes. This has facilitated the dialogue between apparently heterogeneous experts with a positive impact on the interpretation effort. The experts involved come from all continents of the world. This has entailed some organizational difficulties, such as setting the time for the teleconferences, among others, but overall these have proven to be solvable. There is a need for accurate, standardized, and reproducible functional assays that ideally should be applicable on a large scale. The interdisciplinary collaborative approach undertaken by Insight has proven very fruitful and can be helpful to solve the discordances contained in the ClinVar database. On the other hand, Insight acknowledges the need for harmonization with the ACMG guidelines and to work together with ClinGel not only on Lynch syndrome but also on other hereditary GI cancer conditions to provide a consistent classification approach for these genes. There is a need to increase automated interpretation and software tools, including data mining, are being developed to this aim. Importantly, funding is a prerequisite to achieve systematic classification. The original HMPCC database began spontaneously and was sustained by the enthusiasm of the scientists who curated it. With time, without specific funding, keeping up with an increasing amount of data became more difficult. Thanks to the efforts of Finlay McRae, Insight has been able to attract specific funding for the curation from different sources, the latter one being the Colon Cancer Family Registry through an NCI grant, which will allow interpretation of a large number of variants coming from this source. And of course, experienced and competent biocurators are essential to achieve the aim to perform accurate classifications and ultimately to help patients and families. Before concluding, I would like to acknowledge the contribution of the Insight Week, of which you can see here several members. The list would be too long, so I will only mention a few key persons who have also contributed to this presentation. Finlay McRae, the main inspirer of this initiative since 2007, John Paul Plazer, curator of the database in the last decade, Elke Holinsky-Feder and Stefan Aretz, who chair the MMR and APC VIX respectively, Bryony Thompson, and Mark Greenblatt, who is also a member of the ClinGen expert panel and is promoting the harmonization of the two initiatives. I would also like to thank Sherry In, who has provided the APC data presented in this talk. And finally, thanks very much to all listeners for your attention.